I'm sure that's a good thing. Bad hair week.
Bad
So
hair.
my printer is broken, so I'm going to be talking with extra pauses today.
Oh, that's
So I'll have my
fantastic
stories
for a vibrant,
of
dynamic
Yeah, interesting cause and
radio
effect.
program. I know.
reading off of a screen. Scroll.
Okay.
You can things side by side windows.
More monitors.
Yeah, more monitors.
More
I've got two going at the moment. I left my other computer in the other room. My laptop is in the other room. I decided to leave it at two computers this evening.
How'd this work? If I lean over here, Kirsten can actually reach out. You got to reach the other way. Reach out.
I want to do a test here. This is only a test. Don't get excited now. This is just a test.
Yeah. I'm already excited.
We like the test.
All right, so you guys all heard that.
Yes, we did.
So
That's so
I
exciting.
will be able to play that music for you when you need it.
Yay. So that'll be right after. Justin, you have a disclaimer, yes?
I do. Are you ready for it?
Well, are you guys ready? Because I can push some buttons and make everything happen.
Make it go.
So the music comes after the disclaimer. I see it. It's actually what it says right in the notes.
I put everything in the notes, John.
I love it. I love it. Okay.
I try and organize.
Notes?
Yes, the show notes, Justin.
Everything
Here.
except Justin's segments. All right. We will miss Allie, but the show must go on.
I know. Allie is sick tonight. We hope that she feels better. The show must go on.
Okay, I'm gonna give it over to you. When you
Awesome.
want to start, give me a countdown.
Thanks, John. It's all you, Justin.
All right. 3, 2, 1. Disclaimer. The following hour programming is leaking information at a tremendous rate and will not be contained. While the exact size of the informational flow is unknown, the best estimates by experts of information are considered to be greatly underestimated, at least according to experts on estimating, who contend that most experts on information tend to overcompensate for their natural tendencies to overestimate by grossly underestimating. While the size and scope of the eventual impact of the programming is currently being held within oil executives' hair of becoming a global disaster at the surface, the deep undercurrents of scientific knowledge are sure to slick mental shores far and wide before the hour is up on This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good evening, Kirsten.
Hey, Justin. How are you doing this week?
Woo! Doing awesome.
Woo!
Except for my
Awesome.
printer.
I know. Printer technology damage. We need to get you a consistent method of printing. I'm thinking, we can get an ancient printing press, maybe. All the little letters.
The printer's fine.
Stamp.
The printer's fine. It just, something got messed up on my computer, and I think I got rid of the driving or something, and it'll work there later. But it had to break right. This is the only time I use the printer. So the only time I know that it's not working
Is right before the show.
is right as I'm printing, which I never do, but like 10 minutes ahead of either leaving the door or turning on Skype. So
I love it. I love it. Well, welcome, everyone. It's This Week in Science. We are here once again to talk about a whole bunch of science that happened this last week. And as usual, so many stories had to flow right past us, even though I was reaching out and grabbing them and thinking, oh, I want that story. I like that story. And we only had to choose a few, unfortunately. But what's on the show today? I have stories about chemical dino birds, DARPA's lab on a chip, and stem cells for hearing. What did you bring, Justin?
What do I have? I have steroids are good for you. I have a great. My favorite story is this as a mouse fear story. What else we got? We've got knife wielding robots. Toxoplasma gondii on the move. And what is this one? That's the same one. And beneficial bacteria. One of my favorites.
We like the beneficial.
And then a B list that's even as interesting. There's a ton of great stuff out there this week.
I know. There were so many stories, and I love them all. But to get started, my big story, because I'm a bird fan. People who've listened to the show for a while know this about me. So, of course, the birds win out over all others. And this is actually a really cool story. I mean, researchers are using X-rays, high-powered X-rays, to examine dino bird fossils and connect them to modern birds. So, from the story that was sent to me by Pamela Sue Taylor, first quote of the story: Archaeopteryx is to paleontology what Tutankhamun is to archaeology. It's simply one of the icons of our field, said University of Manchester paleontologist Phil Manning. You would think after 150 years of study, we'd know everything we need to know about this animal, but guess what? We were wrong. And that's pretty much because researchers have never looked at Archaeopteryx in this particular way before. Using the X-ray beam from the Stanford Synchrotron radiation light source, which is located at the U.S. Department of Energy's SLAC National Accelerator Laboratory, scientists peered at the chemical remains of the dino bird fossil. They recorded how the X-ray beam interacted with the fossil and created a microscopic scale map of chemical distributions. The chemical map was published in the Proceedings of the National Academy of Sciences this last week. People, quote, have never used a technique this sensitive on Archaeopteryx before, said SLAC physicist Uwe Bergman, who led the X-ray scanning experiment. Because the SSRL beam is so bright, we were able to see the teeniest chemical traces that nobody thought were there. So the scanning process found about a dozen chemical elements that were a part of the animal specimen rather than the surrounding soil that the fossil was embedded in. And this was determined by comparing the relative distributions of the chemicals and finding significantly different concentrations in different places. So in the fossil remains versus in the soil around the fossil. And so this confirmed that the elements probably didn't leach from the soil into the fossil, but were actually most likely part of the fossil all along. The chemicals they found provide a solid link between the fossil dino bird and today's birds. The ancient feather impressions were high in phosphorus and sulfur, two chemicals found in feathers of birds today, and the fossil bones contain copper and zinc in trace amounts. Again, that's really similar to what's found in the bones of modern birds. University of Manchester geochemist Roy Wogelius, he's a corresponding author to for the paper, so if you want to talk to any of the authors, this is the guy you want to talk to. He said, we talk about the physical link between birds and dinosaurs, and now we have found a chemical link between them. So there's been the physical link, and now they have an actual chemical link to solidify the relationship even more. He also says, in the fields of paleontology and geology, people have studied bones for decades, but this whole idea of the preservation of trace metals and the chemical remains of soft tissue is quite exciting. We were able to read so much more into these organisms now using this technology. We're literally touching ghosts, says Regelius. I know. Chemistry is the real key in the future of paleontology. This is a paradigm shift.
This is going to be totally off base. Part of it, because we have a video version of this show now for anybody who's just listening to radio. But now, when I make a sound like like there's this whole cutaway where it's just me after I've made the sound, and it's like, well, it wasn't. No, I didn't have anything more to say than that. Like the editors are all really ready to jump away to another cut shot so I can have a reaction, something. But it was just me making a sound.
We like you having reactions, Justin. It's okay. I don't know. I think this study is really cool. Just the fact the more advanced modern technology gets, it just allows us more and more ways of looking at the bones of these ghosts from the past, you know?
Or the bones of the earth even. They did there was another study, that's one of the B-sides,
Mm,
where they were looking at silver. And some of the components in silver, I guess, were indicating that in the early stages of the Earth, there had to be a certain amount of water present. So
huh.
even though there is this whole theory of comets bringing large amounts of water to create the oceans and the atmosphere, according to these metals and the way they're configured, there must have been water present in the early stages of the Earth. So
Oh, that's interesting.
yeah, there's a whole bunch of ways of looking at the universe that can tell us cool stuff.
I know. And I love the idea though that we don't we don't know how we can look at something until we have a new way to be able to look at something. Whereas before it was always, oh, we're going to touch the bones, we'll, wipe them off with a little brush and look at what the characteristics of the bones are, and oh,
And it
let's
was
cut them in
morphology.
half. Yeah.
Morphology, which is literally you look at one set of bones and try to imagine how it morphed from there into something else. And that's how you designated where something was along an evolutionary path and where it fit into species and everything else. The more similar something was, the more it was put into the same category. And more through the genetic testings, we're finding that they're not always accurate.
Yeah, and that it just goes to show that so often we put these use the physical characteristics to create these relationships, these phylogenetic, but they're not really genetic yet, relationships. And then you need the genetics, you need the chemistry, you need these different ways of understanding how different species ended up evolving, morphing into something different. How did they get there? Why? I mean, knowing that the bones of Archaeopteryx contain the same trace amounts of the copper and zinc, it makes you wonder, okay, why is it important for birds to have those in their bones? Why was it important for Archaeopteryx to have it in their bones? When did that originally start, the incorporation of those trace metals? And then the phosphorus and sulfur in the feathers, when did that become important? Was it part of the dinosaur skin as well? And we just haven't looked at that yet. So, I mean, there's all sorts of new ways they're going to be able to look at stuff. It's pretty cool.
Cool. Speaking of evolutionary things, mice.
Nice.
Mice are naturally afraid of cats. Ask any mouse; they can't stand them. They're also afraid of snakes and rats. These are
I'm kind of afraid of snakes myself.
Yeah, I'm afraid of anything that could bite me. So I'm afraid of cats too, and even mice. It doesn't matter. So so they're so afraid of them that just getting a whiff of them, just sniffing one in the air, is enough to get their hormones pumping, their stress hormones going through the roof. They start to go into high alert mode, looking out for the predator just from smelling them. So the question is, why? Why would they be afraid of the smell? Now, to you and me, it may make sense, just logically. If we were
I don't want to be eaten.
Yeah, if we were shrunk down to the size of a mouse, we might be afraid of cats and snakes, even more so. Can you imagine being the size of a mouse and then a snake comes by, right? But we're doing this as logical sentient beings, and so the question is, how does a mouse know to be afraid? Because most instances, they probably haven't heard from other mice, we would assume. They haven't been told by other mice to be afraid of a snake. They haven't seen snakes eating mice on television. They haven't read up about what snakes like to eat. So why are they afraid? Why are they afraid of cats? Why are they afraid of snakes? Why are they afraid of rats? The answer?
Yes.
New research reported in May 14th issue of journal Cell is discovering that the triggers for the instinctual fear could lie in species-specific urinary proteins known as MUPs, which are secreted by almost every land-dwelling vertebrate. But this is the really amazing thing to me here, is that it is species-specific proteins. So it's not going to be the protein from a human. It's not going to be the protein from a bear, but it'll be a cat, a rat or a snake, those that are predators.
But in their urine, it's protein in their urine. So wherever they go to the bathroom, then if the mice walks around there sniffing.
Wherever they go to the bathroom, but it's a molecule. So it's also, it could be anywhere. It's going to be on the cat and the rat, and it's going to probably be detectable lots of places that the cat. Right.
Right. Especially if you're dealing with animals that are territorial, and mark their territory, there's probably, a fence, like a big area that's just filled with that protein marker in the air everywhere.
So these chemicals are picked up by the sensory neurons found in the mouse's vomeronasal organ. Notably, MUPs sensed by this organ were already known to act as chemical pheromones. They're already known to be a way to communicate from within a species. So that the example given here is that these MUPs that are emitted by male mice motivate more aggressive behavior in other males. So it's generally been known that this is a way that the species communicates within itself. The next part, though, is that something called cheromones, which has mainly been identified in insect communication and wasn't known to be used by mammals, which is actually does communication between different species. So now it's not just within the species that's identifying that they have a similar, a similar molecule but this is a completely different species communicating something. So the quote from the researchers: "At first I was skeptical," and this is from Lisa Stowers of the Scripps Research Institute. "In the earlier studies, her team identified MUPs as the active ingredient in the pheromones, but here we looked for the cues for fear and ended in the same family of proteins. So that which was being used to communicate within the species now is generating the fear of other species." This is the really exciting thing about this is that it makes me wonder what came first, because evolutionarily, these mice that they have been raised in a lab since the 1930s, maybe. They've been removed from nature. They are not witnessing other mice being eaten by cats or rats or snakes. They're not, they're evolutionarily, in a sense, a step removed from these predators for a long period of time. And yet the fear is hardwired in there. The trace sense of the urine is triggering the intense fear. Now, when they did something where they blocked it, let's see what they did. I think they had a rat that was that didn't have the MUPs, that was removed of whatever excretes the MUPs, and the mice were not afraid of it at all. In fact, one of the mice went and slept next to the rat.
Aww.
The quote from Stowers is, "We think it was cold."
That's one good reason to go cuddle up with a rat, I guess.
And actually, let me take that back. It wasn't anything they disabled in the rat. It was they disabled the vomeronasal organs of the mice, and then they were interacting with this anesthetized rat. So the question, though, to me, is like, what is this a learned? I think this is a genetically learned behavior. I don't think natural selection allows enough chance for them to have just selected to be terrified of their predators. It seems like at some point this was something really necessary that the body adapted to. But in a way, it has to have happened through some sort of epigenetic mode at that point. I mean, this is me speculating, obviously.
It doesn't necessarily have to be epigenetic if these animals have evolved together over, how long, and rats and cats and mice and snakes have had predator-prey relationships, and they've had, they've had territory or habitat relationships that have been going on for generations after generations and after generations after generations, so long that it could be. I mean, this is the kind of thing, these instinctive fears these are things that are hardwired after a while, and you don't
Right.
have to learn it during the point of time period of time that you're alive for it to take place.
Right. You don't have to learn it while it's here. But what I'm talking about is, where does it originate from? Like, how does that initially get hardwired in? And I think it's an interesting question because it suggests one of two things. It suggests there were lots of mice that were being born that were genetically not afraid of rats and snakes just by pure chance. The ones, they all were eaten.
Exactly. They
And
were not
the
successful.
only ones that were left were those that, when they would smell a cat or a rat, became terrified.
Mm-hmm.
That's one way that it could have
Yep.
taken place. The other way is that the
Highly
genetics
likely.
could have learned. Something being alive today had a bunch of close encounters or was constantly running from rats and cats and snakes, and its offspring were pre-wired to be afraid of them. That's the interesting question, whether or not the body and the genetics can actually learn. And we've seen, and it's not.
So the experiment that would fix that. So what you would need to do is take mice and rats, mice away from the environment. I mean, all of these mice that you're talking about have lived in an lab environment where they're
70 years.
not, they haven't been exposed. So basically, they've already done this experiment. The fact that after 70 years of mice being bred generation after generation and maintaining this fear goes to show that it's probably not epigenetic, and that it's probably something that is from way back in the old days, and it was evolved over many generations of natural selection. And it probably was, those that smelled the cats and smelled the karyotype, or the the karyomone, that they were the ones that were able to escape earlier and survive. They've already done the test just by having all those mice separated for so long. I think. Anywho, let's move on forward. What did I have? Oh, I had a study. DARPA had has something interesting going on.
Am I back?
Did you
oh
go
my
away?
gosh, that was so scary. This just went to complete darkness.
You're back.
Wait. I want to finish up the last story real quick. I think the experiment. I think epigenetics could hardwire for all future generations. I think that's how the hardwiring could form. The experiment I would like to see is to take those same mice and put them near like grizzly bears, or put them, some sort of South American mouse up near a polar bear, something that wouldn't have been around it. Because if there's a predator out there that would eat them, it's not something that's just in predators, or it's not something that's just in general a threatening signal to the mice. Because for it to be as specific as a few species, I think it would have to be learned. But anyway,
yeah,
you're talking DARPA.
I'm going to talk DARPA. Yeah, moving on from
robot cleaners.
Moving on from mice to labs on a chip. So according to this study was mailed to me by Peter Fraser, and the study was written up in popsci.com, but originally it was written up in Wired Magazine's Danger Room blog. And that blog was reporting that DARPA is funding the development of a biosensor to detect whether or not people have been exposed to certain viruses, to the tune of $19.5 million. So they're investing some money in this. In a sense, the sensor will know if you are sick before you do. It's headed up by the director of Duke University's Institute for Genome Science and Policy, Dr. Jeffrey Finsberg. And the research team first identified 30 genetic markers that are activated by viruses in a person. So when a person has crossed paths with a virus, the level of activation of these markers changes in such a way that it's possible to determine viral exposure hours or even days before the person starts to actually exhibit any kinds of symptoms. So this is like early in the process of the body's reaction. And so this is going to make it possible for doctors and scientists to catch and hopefully deal with viral illnesses in a person before they become contagious. And on a large enough scale, this could actually help the medical community deal with or derail an epidemic before it gets started. Traditionally, we've diagnosed these conditions by testing for the actual pathogen, but that's a slow process and it's not effective until you're already symptomatic. Ginsburg told the Danger Room to look at the actual host response instead is a really novel approach. So in human trials that were started last year, 80 subjects were exposed to 3 viral strains, a rhinovirus, influenza A, and the respiratory syncytial virus. And then urine, blood, and saliva were collected and analyzed for the genetic markers that they'd already identified. The results so far confirm that different genes are activated in response to viral infection than bacterial infection. And so this kind of information could actually help doctors minimize the prescription of unneeded antibiotics, which would be very cool. If you just took a little blood or urine or saliva even is the easiest, and just put it on a little tiny plate and then stuck it in a machine and got your answer as to whether or not the genetic markers are there, a doctor could easily say, okay, you've been exposed to a virus or you've been exposed to a bacteria and know which way to go. And it would be much faster than actually having to do the plating that takes place to grow bacteria to see if you've actually been infected by a bacterium. So according to a Duke University press release from last year, biomedical engineers in Duke's Pratt School of Engineering have already designed a prototype of the device that can read the genomic signatures of infection. And over the next 2 years, in the second phase of the study, which they're now in, researchers are going to refine the probe and further validate the genomic signature of infections by additional pathogens, including the seasonal H1N1 virus. Ginsburg says, we expect to gather valuable data about the novel H1N1 virus from these studies. Pre-symptomatic detection of a cold or flu would be a significant advance in maintaining the health of our troops and will certainly be a breakthrough for the public health and well-being as well. Additionally, in a study started in 2009, researchers at Duke are enrolling students who live in the dorms to investigate the onset and the spread of respiratory viral diseases in close living quarters, which we all know dorms are very much close living quarters. The students are asked to report about their health on a daily basis and provide occasional blood and urine samples. So if you're headed to Duke this fall and living in the dorms, well, you might consider taking part in this study. It's good for science.
Yeah, they've actually come quite a long way in the laboratory settings, because it used to be they would have to do all these test tubes, 30 test tubes on one sample from a patient to see what grew there. And they'd have to leave it in there for 24 or even 48 hours to see something growing in a significant quality and then put it under a microscope and try to actually identify the strain. Through gram tests and everything else. Nowadays they do have a pretty cool computer. It's basically they take the sample and it ends up getting routed through a little card like about yay big, right? Mm-hmm. And in it's got lots of little windows, and it goes into the computer and it gets read. I think it's like 6 or 8 hours or something like that, and it goes through and does all of those tests, and then just prints out the answers to what it is. And if there's anything that's questioned or not 100% sure of what it is, you can actually put a syringe into one of the little cubicles of this card, pull the sample out, and retest it somewhere else, or try to grow it if you want to. But to be able to actually do it with while for the doctor to actually be able to get that information in real time would be incredible.
Yeah, it's just the advancement to allow this kind of analysis of fluid samples, of personal body samples, to see if people have been exposed to something. And to have it happen so quickly and easily is amazing now. Like you said before, people would have to pipette each of those test tubes with the 30 samples. It wasn't done by computers. It was done by hand. It was done by people. And now there are robots that take care of this stuff.
Now robots have taken that job.
Yeah. Did you have a robot story? Were you going to bring that up?
Oh, let's see.
Since we're talking about good medical robots, it seems like a good time to bring up.
Yeah, this is fantastic. This is from the BBC via Minion Hinckley, who emailed it in to me. 3 researchers at the Institute of Robotics and Mechatronics at the German Aerospace Agency performed a study to see what perils might arise in a future in which humans and robots work side by side in the kitchens of tomorrow. More specifically, they looked at what sort of dramas could come from giving robots knives and then getting in the way. They used a robot arm that held a variety of bladed tools and programmed them to strike substances that mimic soft tissues. In some cases, the researchers found the robots managed to accidentally inflict wounds that would prove lethal. According to this is a quote from Minion Hinckley, who's reporting, they claim this research is to make robots safer, but they're basically teaching a robot how to stab people.
Yay, accidental. It's quote unquote. Yeah. I hope they have good learning AI in there. Yeah.
Well, it's sort of learning AI, but what they're actually working on is the safety mechanisms. So it's constructed. This whole test was constructed to see if a prototype safety system could limit the amount of damage done by sort of giving the robots sort of like a sensor on a garage, not a garage door opener, although those have them too. But like I'm thinking the side passenger door on a minivan, those electric doors, right? So you hit the button and the door's closing, and if you put your arm in the doorway, just like an elevator door too, you put your hand in the way, it knows there's an obstacle, and it just opens again. So the idea is, but the tricky part is it has to identify the type of substance it's cutting into, because if it's designed to be cutting vegetables, certainly the flesh will feel wrong. If it's designed to cut pork or chicken or some kind of meat, then maybe the distinctions are going to be a little tougher.
Meat is meat.
The test itself involved a robot arm weighing 14 kilograms. And for those not familiar with kilograms, 14 kilograms is 14,000 grams.
Thanks, Justin.
You're welcome. And they gave him about a meter's reach. And it was equipped with a variety of bladed household tools, including a steak knife, kitchen knife, scissors, and a screwdriver. Robot was programmed to use the bladed tools to stab and cut at a silicone lump. Which is basically, that is how they see us. Silicone lumps. Great. They also used it on the leg of a dead pig. And according to the article, they used it on the arm of a human volunteer.
Ah! I want to know who decided to volunteer for that.
It's Germany. There's probably people who are into it. Want to be cut by robots. I
like to be cut. It is.
I like to be cut by the robots. Now watch my monkey dance. So the injuries were significantly reduced. They did a bunch of deep cuts. Most cases, researchers said they could prove lethal if inflicted on an actual living human being. The injuries were significantly reduced when the prototype collision detection system developed by the researchers was turned on. The system uses torque sensors to spot when it's hit different substances and halt movements. It was used to limit damage when... This is the other line. It kills me here. It was used to limit damage when human subjects were tested. not to avoid damage on the human
just
subject.
Limit.
Just going to have less than lethal, apparently.
The big thing here, though, is like really robots, the Asimov laws. Robots are not supposed to hurt or kill humans, but how do they know what is a human? I mean, we have to teach them what is human and what they're not supposed to hurt or harm. And so if you can't get them to figure that out, then maybe they shouldn't be in the kitchen.
There are studies that have looked at preventing big robots from bumping into or falling down on people, tripping over people, crushing them, basically. This is thought to be the first one that was actually testing whether or not they would stab us to death, whether they're going to be shankers. So The results of the study were first presented in early May of 2010 at the International Conference on Robotics and Automation held in Alaska. The details of that get together are a little bit sketchy, as there were no human survivors.
I think you just made that part up.
I might make it up. Now is the part where we dance.
Now we dance. Actually, it is time for us to go to a break. So if you'll stay tuned for just a few moments, This Week in Science will be back after these messages.
And we are back with more This Week in Science.
No, you're not reading the show notes.
We have show notes?
Duh. Just keep quiet for a second. We have an Audible ad, and then the music's going to come back, and then we keep going. Okay? TWiS would like to thank Audible.com for sponsoring this episode of This Week in Science. Audible.com is the leading provider of audiobooks with over 75,000 different titles in a variety of different genres, from nonfiction to fiction. We've got science, which TWiS is very excited about. TWiS has found many science-based books in the Audible library. So take our pick of the week, for example, which is Bad Science by Ben Goldacre. You can start a free trial today and get the TWiS pick of the week or any other audiobook download for free. All you have to do is sign up at audiblepodcast.com/TWiS. And Bad Science by Ben Goldacre is a wonderful book that goes around finding all the bad science that's out there, things that people in the media are saying that just are not true. Things that have been done poorly and those modern myths that are out there, and he kind of breaks them apart and breaks them down. It's a really, really great read, and so I hope that you will join us, because it's going to be June's TWiS Book Club Book of the Month. So you can get signing up with Audible.com for this free audiobook download. You can get started with our science book of the month and get a head start on everybody. So if you go to audiblepodcast.com/TWiS right now, you can get your free download.
And are we back?
We are back.
We are back with more This Week in Science.
Woohoo! Science. I'm excited about the science. I always am. I have a great story that was sent in by David Eckert and Ed Dyer. Multiple people sending in the same story. It's a good one. This one is related to hearing. Is there a stem cell fix for hearing loss? Let's find out. Dr. Stephan Heller, he's a professor of otolaryngology at the Stanford University School of Medicine. He came up with an idea about 10 years ago that may actually be finally seeing results. He hypothesized that stem cells could be used to create a specialized type of cell for hearing called the hair cell. Hair cells live in your inner ear. Like for me, they'd be actually way inside past these headphones. And they respond by initiating a nerve impulse when air vibrations, sound waves, bend the hair cell's tufty hair-like projections. And these projections are called stereocilia. Heller thought that using stem cells to grow hair cells in petri dishes would allow all sorts of development in our understanding of the molecular basis of deafness. So he set to work. And in his research, which is just published in the journal Cell, he details how he was able to make his idea a reality. He says, "We looked at how the ear develops in an embryo at the developmental steps and mimicked these steps in a culture dish. The research team coerced embryonic stem cells from mice and mouse fibroblasts, induced to act like stem cells, through the proper stages of development to eventually result in hair cells. So the cells were first turned into cells of the ectoderm, or the embryonic layer that eventually turns into all sorts of cell types like skin and nerves and stuff on the outside, outer layer. Then they were morphed into otic progenitor cells, and otic means ear, so they're a cell type that begets the ear. And finally, the cells were told to become hair cells. And each step of this process was dictated by a specific chemical instruction that was determined by the researchers. They would bathe the cells in this special bath of molecular chemical instructions, and the cells would do their bidding. Eventually, the researchers made hair cell-like cells in a petri dish. And Heller says, these cells have a very intriguing structure. Yes, intriguing. They look like they have hair tufts of stereocilia. Hey, that's what they're looking for, right? He says, we knew it was really working when we saw them in the electron microscope. They really looked like they were more or less taken out of the ear. So the cells looked like hair cells, but do they work like them? The researchers tested the cells by vibrating them mechanically and looked for evoked electrical signals or potentials. And the results were very similar to what is recorded from hair cells that are actually taken from an ear. Our study offers a protocol to generate millions of functional hair cells from a renewable source, Heller says. We can now generate these cells and don't have to go through dozens of mice for a single experiment. This allows us to do molecular studies with much higher efficiency. That's right, higher efficiency and less killing of animals. Thumbs up, double thumbs up. It gets around a problem for hair cell research, which is scarcity. There aren't that many hair cells available for studies, seeing as you have to get them from animals or from people, and how are you going to get them? So being able to produce hair cells as needed will allow research in this area to totally flourish. David Corey, he's a professor of neurobiology at Harvard University and not involved in the study. He said that it gives a real hope that there might be some kind of therapy for regenerating hair cells. And it could take a decade or more, but it's a possibility. The next steps are going to be studying these mouse hair cell-like cells in more detail and then trying to develop human ones. So, and the big thing about hair cells is the fact that they don't, you don't get more of them. So the reason that a lot of hearing loss occurs from standing in front of, big speakers and loud music and all that kind of stuff is that damage to your ears kills the hair cells in your ears and they don't grow back. They just die off slowly. So being able to potentially regenerate them would be an amazing, amazing step forward.
I don't know. I kind of think that I've been here all along. Cool.
I'm not believing it.
I think one of the things about hearing loss as you get older is I think it's by will. I don't think it's accidental. I don't think it's from too much loud noises over a lifetime. I think at a certain point in life you're like, I just want to hear less of the young people's music blaring from the stereos of the cars that pass by.
Okay, Justin.
I think it's by design. The older and grumpier and grouchier you get, you don't want to hear all that noise of the world out there. Enough. Yeah. Amazing though. Actually, the ears are pretty strong because I thought I would have like some pretty good hearing loss at this point in life, you know. Actually, I thought because there was all the garage punk bands, and then going too close to the speakers at the club, you know, and all that stuff for years.
Yeah.
And then you come home and then your ears are ringing, that high pitched tinnitus thing going on but I can still hear just fine.
Yeah, I mean, there's a certain amount of damage that your ears can definitely take, but after a certain point, and after repeated exposure to those kinds of environments, things are going to go. Things are going to
Go
go
out, kids.
down.
Go out to the music clubs. Go out to the live punk rock shows in small garages. Don't put your earplugs in. You don't need them. What's going to get your ears? What you're going to get your ears is children. Is you're going to pick up your child who's about to cry, and when they get right up here next to your ear and they just full volume scream, that's where your hearing actually goes. That's why adults typically don't have that high end hearing. It's from screaming children. It's not because they listened to too much rock and roll when they were kids. Go out and have your fun. Don't listen to the old crotchety people telling you that you're going to ruin your ears with rock and roll.
And remember the disclaimer that we say at the beginning of every show.
Yeah, don't listen to healthcare professionals. They're probably wrong. We know better.
All right, let's move on to another story.
always makes her nervous when I do that. When I like...
Yeah, because people might actually listen to you. That's what I'm afraid of.
Oh, shoot. Where's the story? Hey, I've been telling people not to brush their teeth for years, right?
Mm-hmm.
I mean, the more important thing isn't that brush, but is sanitizing your mouth. Using
Right. Nope. He's broken up.
Like, did I go? Am I gone?
You're back. Don't move around so much. You're back.
It can't be me moving that's slowing the thing down. It's already slow, and now my moving just doesn't matter. Anyway, that story is not, this one's not the one I've got in front of me. This is a good one, though. This is the University of Leeds coming out with a good reason for us all to get on the juice. It's a natural defense mechanism against heart disease. Could be switched on by steroids that are being, I think, already sold as health supplements, according to researchers at the University of Leeds. University of Leeds biologists have identified a previously unknown ion channel in human blood vessels. Oh, wait, no, okay. There it is. They can limit the production of inflammatory cytokines, proteins that drive the early stages of heart disease. Cytokines.
Yeah.
They found that the protective effect can be triggered by pregnenolone sulfate, a molecule that is part of a family of steroids that are being referred to as fountain of youth steroids because of their apparent ability to improve energy, vision, and memory. Now, again, this hasn't really been long-term tested on humans in any sort of clinical trials, so disclaimer. This could be like when your doctors were giving you cocaine in the 30s and telling you it would give you that extra pep. You know,
Woo!
I don't completely know all the parameters of this yet. But this is interesting. Collaborative studies with surgeons at the Leeds General and Infirmary have shown that this defense mechanism can be switched on in diseased blood vessels as well as healthy vessels. So they can actually use this steroid and in real time see an effect whether you're being diseased or not. Let's see. These are the reason these are thought to be harmless up until this point. They haven't, found any negativity to them yet. It's because they are naturally made in the human body, but the levels of them decline as we age, and rather rapidly they drop off. So there is a market underway to synthetically make these steroids, and they're billing them as health benefits, but they don't completely have the full picture there yet. But the effect that we have seen is really quite exciting and also unexpected, says Professor David Beach, who led the study. However, we are absolutely not endorsing any claims made by manufacturers of any health supplements. Evidence from human trials is needed first. But we're getting much closer to that take a pill, live forever thing.
Yeah, I don't know. Steroids, though, also have downsides. They're not the kind of thing that you want to be taking all the time. I mean, sure, okay, you can take the Fountain of Youth steroids and then you get man boobs.
Yeah. That would be rad. Oh, like, why would I leave the house? Just play with my man boobs all day.
Yeah, these great. Steroids reduce inflammation. You can get these maybe over the counter or whatever in different places. As a result of reducing inflammation, they probably have all sorts of wonderful effects to make you feel better and younger and happier. But you take them every day, and eventually those steroids will turn on you. I mean, this is why there are issues with steroids, well-known issues with steroids. I'm really surprised at this study, actually.
Well, but this is maybe not all steroids are the same though. I mean, this particular family of steroids might, in the right levels, if it's already declining in the body, maybe it keeps the whole thing going. Of course, there's all sorts of hormones and steroids in the body that we need when we're growing, when we're younger, that we maybe don't need when we're older, and could probably facilitate some bad stuff.
Yeah. All right, I'm going to move on to the Minion mailbag, actually, because we have about 10 minutes left in the show, so I figure we can go to the Minion mailbag. If you just tuned in, you are listening to This Week in Science with Dr. Kirsten Sanford and Justin Jackson.
And my pet tiger.
And Justin's pet tiger. Okay. Anyway. From Minion Mark in Michigan, he says, I hear frequent reference being made to the Apple App Store to download the TWiS app. With Android platform phones gradually, and I don't know, I don't think I've ever said to download the TWiS app from the Apple App Store. We say to go to iTunes to the podcast section because that's where TWiS is located in the podcast. But
we have a Droid app, but I don't think we have an Apple app.
Right, which is what Mark we do not have an Apple app. We have a Droid app, and this is what he says: with the Android platform phones gradually picking up market share, please be aware that there is a TWiS app in the Android market, TWiS for Droid. Yes, it is TWiS for Droid, and it was donated to us by Tim Beauchamp, and he is fantastic. He made it all for us, and so if you have an Android phone, you can go to the marketplace and get TWiS for Droid and download our shows to your phone. Minion Mark says, I carry a Moto Droid, but use Dog Catcher to get my dose of TWiS, and I love me some science-y goodness. Getting to my point, a weekly shout out for TWiS in the Android marketplace would be nice to hear. Anywho, keep up the good work and thanks again. That's a really great point.
Android marketplace.
Yeah, so it's a good point. We should tell people, because we did for a while and then we kind of stopped and we haven't incorporated into the nightly outro. But yes, TWiS for Droid in the marketplace.
Absolutely. We're all for the robots dominating.
Yeah. So thanks,
take
Mark.
Mercy upon our fleshy bodies.
So thanks, Mark. Really appreciate the reminder that we need to tell people about stuff like this. Thank you. Okay, so Gary Pabst wrote in and says, I graduated from UCD, that's UC Davis, for those in the know, in 2004 with an engineering degree, but didn't discover you guys until I had moved.
That happens way too often.
People leave Davis and they're like, oh, hey, there's this show there. Awesome.
We weren't a podcast till 2005 anyway, so.
True that. He also says, I would have loved to have listened when I was there, but had no idea you existed. I'm sure things have changed in the last 6 years, but if you don't already, I would suggest some advertising around the science and engineering buildings and everywhere else for that matter. Everyone should have a deeper understanding of science and science news.
That's a very good idea.
Yeah, so I don't know, maybe we'll have to get some flyers and get some student volunteers who we can give coffee or like a bagel or something to go post things around the campus at Davis.
I guess, but
some guerrilla marketing. Maybe we need. I don't know. Maybe we need like something.
Here's the thing, though. If I feel like if we're going around to the science department at a university, The job's already been done. These are kids who found a path into the sciences. These are people who already have some sort of passion for the science. And yeah, they'd be interested because it's cross science and that's great. But I think we really want to reach out to we should go and flyer up some truck stops. You know what I mean?
Good idea.
We should go get some people listening to us who are doing the long haul logging runs or whatever it is. I think we need to reach out to people who aren't already listening to science on a regular basis. That's who I'm more interested in going and grassrooting.
Well, yeah, that's hopefully we can find lots of people who say I don't like science, and then they hear us and we go.
Or better yet, let's go grab a whole big backpack full of science books, right, and bury them in playgrounds. This way the kids around the playground is like, whoa, what's this? They find something buried in the sand. It's like this buried treasure book. Like, what's this book?
It's a time capsule.
And then the teacher's explaining, well, this is explaining the Einstein's theory of relativity. And the kid's like, what's that? And the teacher starts to talk, and then the kid loses interest and goes away. And then 10 years later, they're in the classroom, and then they're hearing about Einstein, the theory of relativity. It's like, oh, I was there. I discovered that. I'm the one that found that. You plant the seed for them to be excited about stuff later on in life. Probably nothing would do that better than finding a buried treasure in the playground. And the teacher has to explain a little bit about the theory, a little bit about the scientist, and then you move on. And 5-10 years later, that seed is already planted. The kid already the name of the scientist, the name of the theory.
Yeah,
that's a real grassroots effort right there.
That's real guerrilla marketing. That's underground.
Yeah.
Underground marketing, PR efforts. I like it.
That's how we take science to the streets.
So Gary wrapped up by saying, thank you so much. I love all the show and all the sciencey goodness. You and NPR Science Friday are the two podcasts I keep up with. Go science! That's right. Go science. Thanks, Gary. Thanks for writing in.
What was the other one?
Great ideas.
What was the other one you listened to? Science what day? Never heard of it.
Never heard of it.
Well, Science Friday does this whole thing on other media that's covering science podcasts. Not once do they mention the show that's beaten them on the iTunes, numerous times over the years. Not even a mention of This Week in Science.
I think it's because Ira is afraid. I don't know. Maybe Ira is worried. I don't know about that. Anyway, we're at the end of our show, and if you haven't started reading already, there is still time to read along with the TWiS Book Club. This month, it's May. The book of the month is Beyond Human: Living with Robots and Cyborgs.
I don't have that book.
Maybe you should get it and read along. It's the book club book. Beyond Human, Living with Robots and Cyborgs. It sounds really interesting. You can get involved in conversations about the book at twisbookclub.ning.com. And I'd like to give shout outs to Gene Culling, Eva Schneider, Mike Klein, and Rob Hinckley for writing in this week. Thank you so much. And for everyone else who sent in stories and all the stories we didn't get to. Hopefully we will get to more during this next week somehow. And I guess that's about it, Justin.
We hope you've enjoyed the show. I don't have the show notes in front of me. We hope you've enjoyed the show. If you liked what you heard, if you have a suggestion for somebody we should interview in the future, drop us an email at justin@thisweekinscience.com or kirsten@thisweekinscience.com. Be sure to put TWiS somewhere in the subject matter, or you will be spam filtered into oblivion.
Oblivion. That's right. And TWiS is also available as a podcast. You can go to our website, www.twis.org, and click on subscribe to the TWiS Science Podcast for information on how to subscribe, or just search for This Week in Science in iTunes. And for more information on anything you've heard here today, show notes are eventually going to be available on our website because it's still there's some back end under construction stuff that's happening. We're going to be rolling out a new website pretty soon, which I'm really excited about.
I don't believe you.
Oh, believe me, there's work happening. It's pretty awesome.
I don't believe you.
Our new site.
I'm going to believe it when I believe it. This is 5 years ago. This is when the show first started, and this is our website. But don't get too attached to it.
I know. Things happen slowly, but they're happening. Totally happening. All right, so we are going to be back here next week, and we hope that you will join us again for more great science news.
And if you've learned anything from today's show, remember,
it's all in your head.
It's all in your head. Nothing going on here now.
Yeah.
All right, kids. Much fun.
That was a lot of fun.
How much fun is this? Look at that. That looks rad.
You and the hussies. You and the hussies.
That's a hussy hat. Can you buy a hussy hat still, Kirsten? Or hussy hats still available?
No more hussy hats.